Welcome to another episode from the Veterinary Research Channel. In today's episode, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024. This research, conducted by Brian Chang and colleagues at the University of Florida, investigates the reliability of several commonly used neurological reflexes in dogs. As veterinary professionals, a thorough neurological examination is paramount in pinpointing the location of a lesion within the nervous system. A key component of this evaluation involves the assessment of segmental reflexes. These reflexes, particularly those in the limbs, often operate through simple pathways involving a single connection between sensory and motor neurons. The patellar reflex, more commonly known as the knee-jerk reflex, and the withdrawal or flexor reflex, which you might observe when a dog retracts its limb from a painful stimulus, have long been considered the most dependable of these reflexes in canine patients. However, the authors of this study point out that there has been a lack of comprehensive evidence to definitively support that these two reflexes are the only truly reliable ones in our canine companions. Several other reflexes, including the Gastrocnemius, cranial tibial, Triceps, Biceps, and Extensor carpi radialis reflexes have also traditionally been classified as myotatic reflexes, meaning they involve that single synaptic connection. Interestingly, the withdrawal reflex stands apart as it requires the coordinated action of motor neurons spanning multiple segments of the spinal cord. Previous research has explored the agreement between different observers when assessing these reflexes in dogs. These studies have indicated that the level of experience of the examiner can indeed influence how well different individuals agree on the presence or absence of a reflex, particularly the biceps reflex. One study even noted that the length of a dog's hair coat could affect the interpretation of the biceps reflex. Furthermore, the classification of the cranial tibial and extensor carpi radialis reflexes as purely myotatic has recently been questioned. Intriguing findings from an unpublished report suggested that these reflexes could still be elicited in dog cadavers even after the major nerves supplying those limbs were severed. This led to another study that demonstrated these reflexes persisted even after a local anesthetic block, raising the possibility that the observed muscle response might not solely rely on the typical reflex arc but could also involve a direct muscular response to the stimulus known as an idiomuscular response. Given these uncertainties and the limited evidence supporting the exclusive reliability of the patellar and withdrawal reflexes, the primary objective of this study was to rigorously evaluate the consistency of 8 segmental reflexes in the limbs of dogs that showed no clinical signs of orthopedic or neurological disease. The researchers aimed to achieve this by measuring both how consistent a single observer was when testing the same reflex on both sides of the dog and how well different observers agreed with each other's findings. The initial hypothesis was that the patellar and withdrawal reflexes in both the front and hind limbs would indeed demonstrate acceptable agreement and provide reliable results in clinically normal dogs. To conduct this study, the researchers carefully selected a group of 101 dogs. These dogs were client-owned or belonged to staff at the University of Florida Small Animal Hospital and were sequentially included in the study over a period of nearly a year. The participating dogs ranged in age from 1 to 10 years and represented a wide variety of breeds, sizes, and sexes. Importantly, all dogs underwent a general physical examination and a complete neurological examination by a neurology resident to ensure they had no clinical evidence of orthopedic issues that could restrict limb movement, nor any signs of myelopathy or neuromuscular diseases or suspicion thereof. Dogs that were too temperamental to tolerate the full reflex testing in a lying position were also excluded from the study. The researchers focused on 8 specific segmental reflexes, 4 in each thoracic limb and 4 in each pelvic limb. In the thoracic limb, these included the biceps reflex, the triceps reflex, the extensor carpi radialis reflex, and the withdrawal reflex. For the pelvic limb, the reflexes tested were the patellar reflex, the cranial tibial reflex, the gastrocnemius reflex, and again, the withdrawal reflex. For each reflex, a standardized technique was used to elicit the response, and the response was simply classified as either positive, meaning the expected muscle contraction or joint movement occurred, or negative, meaning it did not. The study involved 3 observers, a neurology resident and 2 board-certified neurologists. The consistency of a single observer's findings was assessed by comparing the responses of the right limb to the left limb in a subset of dogs. This was done independently by the resident and one of the board-certified neurologists. The agreement between different observers was evaluated by comparing the findings of the resident with each of the board-certified neurologists. Now let's move on to the exciting part, the results of this investigation. The researchers meticulously analyzed the agreement both within a single observer and between different observers for each of the 8 segmental reflexes. They used a statistical measure called the proportion of agreement, which simply calculates the percentage of times the observer's assessments matched. Additionally, they calculated kappa values, a more sophisticated statistical measure that takes into account the possibility of agreement occurring by chance. A segmental reflex was considered to have acceptable agreement if it reached a proportion of agreement of at least 90% and a kappa value of at least 0.61 in both limbs. The study found that the agreement within a single observer was high, at or above 95% for the extensor carpi radialis reflex, both forelimb and hind limb withdrawal reflexes, the patellar reflex, and the cranial tibial reflex across all three observers. When comparing the agreement between different observers, specifically between the neurology resident and each of the board-certified neurologists, they again found high proportions of agreement, at or above 92%. For the extensor carpi radialis, both withdrawal reflexes, patellar, and cranial tibial reflexes. For these reflexes showing very high agreement, the kappa values, when calculable, also indicate a good agreement. In some instances where the observed agreement was perfect or near perfect, the kappa value could not be determined statistically. Interestingly, the intraobserver agreement for the biceps reflex was 94% for both board-certified neurologists but slightly lower at 88% for the neurology resident. The agreement between observers for the biceps reflex showed a high proportion of agreement, but the kappa values suggested only fair to good agreement, and this varied slightly between the left and right limbs. The triceps and gastrocnemius reflexes consistently showed lower levels of agreement both within and between observers. These results led the researchers to conclude that, based on the consistency of positive responses and the level of agreement between observers, the extensor carpi radialis reflex and the cranial tibial reflex, in addition to the long-accepted patellar and withdrawal reflexes, could also be considered reliably present in the study population of healthy dogs. Now, let's delve into the implications of these findings in the discussion section of the paper. The researchers begin by highlighting the crucial role that the assessment of segmental reflexes continues to play in the daily practice of veterinary medicine, aiding clinicians in the vital task of neurolocalization. Despite the inherent subjectivity in interpreting these reflexes, their findings indeed support the long-held belief that the patellar and withdrawal reflexes are reliably present in a population of clinically normal dogs. However, their study goes further by providing evidence that the extensor carpi radialis reflex and the cranial tibial reflex also demonstrate a high degree of reliability in terms of their presence and the consistency with which they can be elicited and interpreted by different examiners. The authors then touch upon previous studies that have investigated the accuracy of the patellar and withdrawal reflexes in pinpointing the location of neurological lesions. They cite a study that found the withdrawal reflex to be unreliable in distinguishing between lesions in different segments of the cervical spinal cord. Another study indicated that a decreased withdrawal reflex was not a strong predictor of lesions in the lower lumbar and sacral spinal cord segments, and similarly, a decreased patellar reflex was not a highly accurate indicator of lesions in the same region. However, that same study found that an increased patellar reflex was more accurate in predicting lesions in the thoracolumbar region. A separate study suggested that a decreased patellar reflex was always associated with lesions in the thoracolumbar region. These findings underscore that while the presence or absence of a reflex is informative, its ability to precisely localize a lesion can be complex and may need to be considered in conjunction with other neurological findings. The researchers also revisit the influence of examiner experience on reflex assessment, referencing earlier studies that found experience to impact the agreement on the biceps reflex. Their own data supports this, as the board-certified neurologists in the study showed higher agreement on the biceps reflex compared to the neurology resident. Additionally, the impact of hair coat length on the biceps reflex, as noted in previous research, is acknowledged, although their study still found the biceps reflex to be potentially reliable despite including dogs with long hair coats. The researchers speculate that inconsistencies in the interobserver agreement for the biceps reflex between the left and right limbs might be due to subtle variations in technique or patient positioning affecting the visibility and interpretation of the reflex response. They suggest that video analysis could potentially help to clarify these subtle responses in future studies. A significant contribution of this study is the classification of the extensor carpi radialis and cranial tibial reflexes as reliable for neurological examinations in dogs. The authors note that the cranial tibial reflex is often easier to elicit due to the readily palpable cranial tibial muscle and the clear flexion of the tarsus that constitutes a positive response. Similarly, the patellar tendon is easily located and the extension of the stifle is a distinct movement. The withdrawal reflex, elicited by a noxious stimulus to the toes, also typically produces a clear flexion response. In contrast, the extensor carpi radialis muscle is smaller and harder to palpate, lying near other carpal extensor muscles, which could lead to misinterpretation if one of the adjacent muscles is inadvertently struck. The discussion then shifts to the nature of the extensor carpi, radialis, and cranial tibial reflexes. The researchers cite a previous study in cats suggesting that these reflexes might not be strictly myotatic but could involve an idiomuscular response, a direct contraction of the muscle fibers upon percussion. While idiomuscular responses are not commonly relied upon in human medicine due to their uncertain mechanism and clinical significance, these reflexes are routinely included in veterinary neurological examinations. The authors caution that while these idiomuscular responses may be easy to elicit, their presence or absence may not directly correlate with the precise location of a neurological lesion. The authors also openly discuss several limitations of their study. Firstly, the number of dogs examined by each of the 3 observers was not the same, which could potentially have influenced the results and the observed agreements. Secondly, while all included dogs were neurologically localized to the brain or had normal neurological exams, advanced imaging to definitively rule out spinal lesions in all animals was not performed, meaning some dogs might have had concurrent, undiagnosed spinal issues. Thirdly, the researchers point out that even when the observed agreement and kappa values were high for certain reflexes, the statistical confidence intervals could be quite wide due to the sample size, indicating some degree of uncertainty. They illustrate this with the biceps reflex, where a larger sample size would have likely resulted in a narrower and more precise confidence interval. For the highly reliable patellar and withdrawal reflexes, kappa values could not even be calculated in some comparisons due to the perfect or near-perfect agreement, highlighting a ceiling effect in the statistical analysis. Finally, the study's external validity is inherently limited as it was conducted on a convenience sample of dogs presenting to a single university veterinary hospital, meaning the findings may not be directly generalizable to all dog populations. In conclusion, this study provides valuable insights into the reliability of several commonly assessed segmental reflexes in dogs. The researchers found that the patellar and withdrawal reflexes in both the thoracic and pelvic limbs, as well as the extensor carpi radialis and the cranial tibial reflexes, demonstrated a high degree of consistency and agreement in their study sample of 101 dogs. Their findings strongly support the continued inclusion of the patellar and withdrawal reflexes in routine neurological examinations of dogs. Furthermore, this research suggests that the extensor carpi radialis and cranial tibial reflexes may also offer valuable information when performing a neurological assessment in canine patients. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.